the patent right for the next 20 years. That means the next 20 years you are the operator of this molecule, you are the owner of this molecule. In the research world, the success of a scientist or the success of a pharma company will be measured by how many patent rights they have. If you have more and more patent rights, you are more and more successful research scientist. If you have more and more patent rights, you are more and more successful pharma company and R&D company. So the next 20 years, you are the owner of this molecule. You only can manufacture this drug. You only can market this one. If somebody wants to manufacture this one, market this one, they have to pay royalty for you. And this called as a drug discovery, but initially it has got only 20% activity. Now what you will do? Somehow you will increase that activity from 20% to maybe 95% or 98 to 99%. You will try to increase that activity. You will purify them, you will isolate them, you will add new functional groups, you will delete unnecessary functional groups so that you are going to increase that activity. If you could able to increase that activity to that maximum level, now this process we call it as a drug development. So that means you develop this drug further. Generally, where we do this discovery and development? Generally, we do this discovery and development in R&Ds. Most of the pharma companies will have R&Ds. In those R&Ds, all these research scientists, they try to isolate, identify the drug, and they try to develop it. Most of the pharma companies will maintain R&Ds. Not only those universities, government research organizations, and the private research organizations also, they try to find out new drug molecules like this one. As an Indian government rule, 20% of your property, your profits need to spend on R&Ds. As a pharma company, you have to spend on R&D. Otherwise, you will not have a new drug, so you are going to lose the market competition. As a pharma company, you have to thrive developing on new molecules. Otherwise, whatever the existing molecule that you have, you need to develop them. Otherwise, you lose the patent right after 20 years. Okay? Now, we do R&D and you completed R&D. Now your drug is ready within your hand. Now we need to prove that whether this drug is working or not. How we can prove that whether this drug is working or not? To prove that one, now I will do the first thing. I will go with the in vitro study. Do you know anybody what you mean by in vitro study? In vitro means using artificial conditions or in lab, or also called as in Petri plate or in test tube. During the in vitro study, what we are going to do in the laboratory conditions, we are going to prove that my drug is very much safe. I need to prove that my drug is not a toxic, my drug is not a carcinogenic, not a carcinogenic. My drug you need to prove is not a mutagenic. You know what you mean by toxic? It should not cause any toxicities, and it should not be cancer causing. It should not be carcinogenic, and it should not be mutagenic. Sometimes the mutations may lead to cancer. So during the in vitro study, in the laboratory conditions, I will do all the toxicity tests, carcinogenic tests and mutagenic tests, and I'm going to prove that my drug is 100% safe. It won't cause any toxicity or nor carcinogenicity nor mutagenicity. Once I prove that, then what I will do? Now I will go with in vivo study. Do you know what I mean by in vivo? In vivo means in natural conditions. What is a natural condition for a drug? The natural environment for a drug is inside the body. Isn't it? Inside a living organism. That is the natural environment for a drug. So that means what I will do during the in vivo study, I will inject my drug to some living organism and I will test my drug in that living organism. So during the in vivo study, what we do, we will select one animal model. In that animal model, I will prove that my drug is safe. My drug is effective. There are different types of animal models are available in the market. Depends upon the drug, depends on the therapeutic area. You will choose any one animal. Since I told you in the starting, my drug is going to work on the diabetes. Remember I told you my drug is working on diabetes. So diabetes comes under which therapeutic area? Anybody? Diabetes comes under endocrinology. So my therapeutic area is just imagine it's an endocrinology. My drug is going to work on a condition called diabetes. So according to that, I'll choose any one animal model. So there are different types of animal models are available. Say for example, rat is one animal. Say for example, we have a mice, we have a guinea pig, we have a guinea pig, we have say for example rabbit, we have a monkey, we have a chimpanzee, horse, sheep, cow. There are different animals. Remember, when the Bharat Biotech working on the Covaxin, they worked with monkeys. And when AstraZeneca worked with the Covishield, they tested this drug on chimpanzees. So it depends on my drug and therapeutic area. I'll choose any one animal model. In that animal model, I'll test my drug. So for example, in today's class, to prove that XYZ drug is safe and effective, I have taken rat as my animal model. Just imagine. So once I have selected this rat animal, then what I did, to test the drug, wantedly, I have removed pancreas from this animal. Just imagine in that way. I have removed the pancreas. Tell me what will happen if you remove the pancreas? If you remove the pancreas, there will be no insulin. There will be no insulin secretion. If there is no insulin secretion, what will become? This animal will become diabetic. It will become a diabetic. Understand this one? Once it has become a diabetic, what I have done, I have taken samples from this animal. I have taken the blood, the serum, plasma, sputum, urine, biopsies. I will take all the samples. Once I have taken the samples, what I did different types of diagnostic tests. All types of tests. When you perform the test, what will happen? You will get the test results or test reports. Now I have collected all the test reports in the form of a table. I collect all that information in the form of a table. If you collect information in the form of table, what do you call it as a name? That we call it as a name data. If you collect information in an organized manner, simply what you call it as a name? Data. Just imagine, what is the test I have performed, what is the result I have got, and what is the indication now, and what date I have performed this test. Just imagine, I did the blood glucose test, okay, and the result is 450 μg per ml. Just imagine like that one. And what is the indication? This is very high, and I conducted this test on 20th of November, 20th of November 2024. And then I did the blood urea test, I did the serum creatine test, I did the cholesterol, I did say for example, hemoglobin, I did say for example RBC, WBC, alkaline pocket. Just imagine, I did 200 different types of tests. So I will collect all the test reports in a form of table like this one. You know already what you did with table, right? It is going to have columns and rows. So if I collect information like this one, now this, what you call it as a name? This we call it as a name data. Now, once I collected the data, after some time, to the same rat animal again, to the same rat animal, this time, what I did? I have injected my XYZ drug. Remember in the starting I told you, I have discovered a new drug called XYZ. I told you, I'm hoping that this drug is going to work on diabetes. So I have injected that XYZ drug. Once I have injected XYZ drug, after some time, again I have collected the samples. Again, I have done the same test that what I did previously. I did the same test again one more time. Again, if you perform the test, what you will get now? You will get the test reports or test results. Again, I collected all the test reports in a form of a table. Again, if you collect information in a form of a table, what you call it as a name? We call it as a data. Now I have two different data. Do you know what is the difference between this data and this data? By the way, I collected both the data from same animal. What's the difference between this data and this data?
After the drug induced.
Very good. This data is before giving my drug and this data is after giving my drug. Now, if you want to prove that your drug is working in this animal, how we can understand this one? By comparing this data with this data, we can come to a conclusion. Say for example, I will write a small example so that you can tell me what is your conclusion on this. Now, before giving the drug to the same rat animal, you know what I did? I did the blood glucose test. Okay? When I did the blood glucose test, got the value 450 μg per ml. Now, after giving my drug, after giving my XYZ drug to the same rat animal, again I did the same blood glucose test. This time, the value is 200 μg per ml. Now, what is our conclusion? What you can say for this? You know, the glucose levels has been reduced from 450 to 200, hence proven my drug is working in this animal. My drug is working in this animal. Now, can I come to the conclusion like that?
No.
Why no?
You're supposed to do it for a small side effects.
You know, the glucose levels has been reduced from 450 to 200, that's fine. But do you think the reduction in the glucose level is only because of my drug only?
No.
We never know.
Sir.
We don't know. We're not 100% sure. Why, you know? Because the reduction in the glucose level or increase in the glucose level from one value to another value, it may or may not depends on my drug. Because so many other factors can also influence the blood glucose level. Isn't it? So, if you want to come to a conclusion, only the glucose report is not enough. We need to consider other factors also, other variables also. What are the other factors? Do you know? Can influence the glucose level?
Sex, diet, diet, exercise.
The gender of the person, the age of the person, the most important
Diet.
the diet. So, what is the food quality that we have taken? How much quantity that we have taken? What is the quality of the food? How much carbohydrates we have in that? How much of fats or proteins we have in that? And it also depends upon the exercise that the patient has been the patient has been done, and the rest he has taken. It is also depends on the cholesterol problem. It also depends on the hypersensitivity. It is also depends on the stress. So many other factors can also influence the glucose level. So, if so many factors can influence, then how can I come to conclusion by only seeing these two values? I cannot come to conclusion. If I want to come to a conclusion, I have to consider all these variables, all these factors. So, if you want to consider all these factors, now what we should do? If you want to consider all these factors, so we have to collect this information also. If you want to collect that information also, what should I do? I should not take only one animal. I need to nullify the gender factor. So what should I do? Maybe I should take 1,000 animals. I need to take out of 1,000, I should take 500 male animals. I should take the 500 female animals. In that male animals, I should take different types of age groups. Maybe 1 month, 2 months, 3 months, 4 months, like that one. And within the different age, I should take different types of weight, different types of body mass index. I should different types of diet I should give. I should give different types of exercise, different types of resting period. I should give different types of stress, different types of cholesterol, hypersensitivity. And while I'm doing all this, I need to collect all this information. Just imagine if you started collecting all this information from 1,000 animals. Just imagine how many lines of data that you are going to get. You are going to get millions of lines of data. You can't even imagine. If the data is maybe 10 lines or 15 lines, you no need to have any help. By seeing the data, you can come to a conclusion. But if the data is 100 lines or 200 lines, at least you need to have a calculator. But if the data is 1,000 lines or 2,000 lines, at least you should have an Excel sheet. But if the data is millions of lines, none of these tools will work. If the data is so huge, how to analyze this data? To analyze this data very fast, with very accuracy, we need to have a software tool. A software tool that can analyze the data with high accuracy within no time. I want to do that one. So, whatever the software tool that you are going to use to analyze this data, and that tool we call it as a data analysis tool. We should use a data analysis tool because the data is going to be enormous, huge amount of data. I want to analyze the data with high precision, without any, errors. I need to analyze that one. I need to analyze it very faster. So I will use some software tool, and that tool we call it as an analysis tool. There are around 20 to 25 different data analysis tools are available in the market. Which tool you need to choose? And by the way, SAS is one among them. And there is a tool called R, there is a Power BI, there is a Tableau, there is somehow we can use Python, SPAS, ClickView, Apache, Spark, JReview. There are around 20 to 25 tools are available. Each tool has got its own advantages and disadvantages. So, say for example, I have chosen one tool, maybe SAS or R or Python or something like that, and I need data analysis. And by doing the data analysis, what I got now, I got the analysis report. Whatever the report I have got, with this analysis report, in future, I am going to fill an application called IND. Anybody knows what is the full form of IND?
Investigational
new drug, very good. Investigational, new drug. So I'm going to fill an application like this one, and I will submit this application to FDA. I'm an Indian pharma company. I want to market my drug in India. Then why should I launch my application to FDA? As a pharma company, if you market a drug in only one country, you won't get good profits. You have to market it internationally. So if you want to market your drug in future in USA, definitely you need to get a permission from FDA. Most of the countries similarly, more or less is going to have the same regulations like FDA. So if FDA gives the permission for USA, getting permission for other countries also will become easy. Because there is an organization called ICH, it maintains an harmonization. So that's why I will launch my application directly to FDA. Now FDA will take 1 month time to review your application. If they strongly believe that your drug was very promising in these animals, it showed good effects with less side effects, better efficacy, then now FDA will give the permission to conduct clinical trials in humans. They'll give the permission now. Most of the pharma companies will have their own animal houses. They are going to have their own people. They'll conduct the clinical trial up to this point. By the way, I told you during the in vivo study, we test our drug on some animals. So that's why this in vivo studies also called it as an animal studies. Okay? But this is before the human clinical trials. That's why these are also called as a pre-clinical studies or pre-clinical trials. Understand this one? Because during the in vivo study, we test our drug on animals, so animal studies. Before human studies, we do this one. That's why it is also called as a pre-clinical studies. Now, as I said, up to this point, most of the pharma companies will do by themselves. But once they got the permission to conduct the clinical trials in humans, now these pharma companies, now they will approach CRO companies. Do you know what do you mean by CRO?
Clinical Research Organization.
Clinical Research or Contract Research Organization. Now these CRO companies, on behalf of the pharma companies, they will conduct the clinical trials in humans. Certain CRO companies, they will only conduct the clinical trials. certain CRO companies, they'll conduct the clinical trials, plus they do the data analysis also. So that means certain CRO companies, they will do end-to-end services. There is a CRO company called, say, for example, Hetero and GVK and a few other small CRO companies. So, what they will do? They will do only clinical trials in humans. But we have a few other CRO companies like Cytel, Paraxel, Cognizant, all these CRO companies, they work only with the data. But few CRO companies, they do end-to-end. So right from the conducting the clinical trials, collecting the data, analyzing the data, submitting the data to the FDA, they will do end-to-end. But there are some sponsor companies like Novartis, Novo Nordisk, Pfizer, GSK, Sun Pharma. These companies, they will have their own product. They will conduct the clinical trial by themselves. They do the data analysis. They will submit the analysis data to the FDA also. Those are called sponsor companies. So we'll discuss about that some other time. So once they got the permission to conduct the clinical trials, now they will approve the CRO companies. Now these CRO companies, on behalf of the pharma companies, they will conduct the clinical trials in humans. Is this clear up to this point, everybody? Is this clear up to this one?
Yes, sir. Yes, sir.
Now, in humans, we conduct the clinical trials in 4 phases. The first one is called as the first one we call it as phase 1 study. The second one is called as phase 2 studies, and then we are going to have a phase 3 studies. Then you will have a phase 4 studies. When you go for an interview as a fresher or 1 or 2 years experience, these questions are very, very important. Can you tell me how many phases that we have in human clinical trials? There are questions in this one. Yes. The first one is the phase 1 study. The main focus of the phase 1 study is safety and dose confirmation studies. Safety and dosage confirmation. So, whether the drug is working or not, that is the secondary. First, we need to prove that our drug is safe. Then how we can prove the safety? To prove the safety of the drug during the phase 1 study, what we are going to do, we are going to take around 30 to 70, sometimes even 100 healthy humans. Some people call it as volunteers or healthy humans or like that one, but you should not discriminate as a healthy or patient. We should call them as subjects. Who is a subject? A subject is a person who involved in a clinical trial. He may be a healthy person, may be a diseased patient, or with a certain condition. So we should call them as a subject. So we are going to take around 30 to 70, sometimes 100. This is not any constant value. It depends on the drug, the therapeutic area, the availability of the subjects. If it's a rare genetic disorder, it's very difficult. But anyhow, in the phase 1, we take only healthy humans, so 30 to maybe 70 to 100. Now, in these 100 subjects, I need to prove that my drug is safe. Then how we can prove that one? Here also same story. To all the 100 subjects, what I'm going to do, before injecting my drug, I'll collect the samples. We are going to run the test. When you perform the test, so you get the test reports. I'll collect all the test reports in a form of data or a table. To the same 100 subjects, again what I will do, I'm going to inject my experimental drug. After injecting my experimental drug, again what I will do, I'll collect the samples, I will perform the test, I'll get the test reports, again with the test reports, I'll collect the data. So that means, again, if you want to prove the safety of a drug, what I should do, whatever the data that you collected before giving the drug, after giving the drug, we need to compare both. Comparison is not very easy. But you have to do analysis and then we should compare. I will take a simple example. Tell me what is your opinion on this one. Before the drug, I have taken one of the subjects from this 100 group. So before the drug, there is one subject ID called 101. To this 101, I did say, for example, blood pressure test. I measured the blood pressure. Okay, when I measure the blood pressure, since he's a healthy patient, healthy subject, what is the blood pressure level of a healthy person? Systolic is 120 divided by diastolic is 80. Since in a healthy person, he got 120 by 80. Now, I have given my drug after injecting my drug. After injecting my XYZ drug. After 1 hour or 2 hours, again, I measure the blood pressure level. This time, the value is, say for example, 160 by 110. What is our conclusion? What is our conclusion? Before giving my drug, it was simply very normal 120 by healthy, 120 by 80. But after giving my drug, now the blood pressure level has been increased to 160 by 110. Maybe my drug is not safe. Isn't it? Because there is an increase in the blood pressure level after giving the drug. So maybe my drug is not safe. But can I come to conclusion that much easily? No. Because the blood pressure, increase in the blood pressure of any patient or a subject may or may not depends upon my drug. Because the blood pressure level influenced by so many other factors. It can be influenced, isn't it? Again, it can be influenced by the gender of the subject, the gender of the subject, by the age of the subject, the weight of the subject, the body mass index, right? Again, the cholesterol problem, again the stress, hypersensitivity, isn't it? Again, the thyroid, or maybe some nutrients, say for example, sodium, or maybe potassium, or some other maybe. We never know. So, again, if we want to come to conclusion, again, what we need to do? We have to consider all these factors. Okay, if there is one student, he got 100 out of 100, so therefore he is very hardworking. Can I come to conclusion that much easily?
No.
May or may not, isn't it? Maybe hardworking, may not be, maybe smart working. Or maybe lucky, whatever questions that he prepared, he may got maybe he got the same questions. We never know. So, if you want to come to a conclusion, we have to consider so many other factors also. Again, if you are considering the amount of the data that you are going to collect will be huge. So what I want to say, at the end of the phase 1, you are going to get a huge amount of data. So you have to focus majorly on the data. Now once you complete it, by the way, during the phase 1 itself, we are going to prove maximum doses that we take without side effects. That is called as a maximum tolerable dosage. We call it as an MTD. Maximum tolerable dosage. You know how we are going to prove that one? In this, we were going to take, just imagine we are going to take 3 subjects, subject A, B and C. To all the 3 subjects, just imagine I have given 50 mg, 50 mg and 50 mg dose. After giving the 50 mg, I have, observed for if anybody has got any adverse event side effects. Nobody has got any side effects. Then after that, I have increased the dosage level to 100. After giving 100 also, nobody got any side effects. Then I have increased that to 150. After increasing that to 150 also, nobody has got any side effects. And then I have increased it to 200. And after increasing the 200, one of the 3 subjects has got, say for example, vomitings. One of the, patient subject has got vomitings. I will stop there. I will go back 1 step again. And again I have given 150 to all the 3 subjects. When I have given 150, nobody has got any side effects. And again, I came back to 200. When I give the 200, this time, one of the subject is started showing allergic rashes. Then I will stop there. I will stop in this case, in this step, and then I am going to say 150 is the maximum tolerable doses that we can give without any side effects. This is how we are going to determine the maximum tolerable dosage without side effects. And then based on the disease severity, I may give 50 mg or 100 mg or 150 mg. Later I will test that different dosages. So in the phase 1, what is the main focus of the phase 1 study? The safety and maximum tolerable dosage without any side effects. Understand this one, everybody? Now, once we completed the phase 1 study, then we will go with the second one, which is a phase 2 study. Okay, we'll go with the phase 2 study. You know, the main focus of the phase 2 study is efficacy of the drug. In the phase 1, we have proven safety. Now we are going to look into the efficacy. To prove the efficacy, you know, among which humans we can prove the efficacy. If you want to prove that whether my drug is working or not, what kind of humans I need to select here?
Disease prone.
To prove that efficacy, during the phase 2, we are going to select disease patients. Remember in the starting I told you my drug is going to work on diabetes. So that's why in phase 2 what I need to do? I need to select only diabetic patients. I need to select only diabetic patients. So during the phase 2, we are going to select maybe 300 to maybe 700, sometimes even 1000 diabetic patients. If my study is a migraine study, I need to select only migraine patients. If my study is a cardiac study, I need to select only cardiac patients. If my study is some thyroid related, I need to select thyroid patient. If my study is an asthma related, I need to select only asthma patients. So it depends upon our study disease. We are going to select the patient group. And as I said, we should not discriminate them as a patients. We should call them as subjects. In these 300 to 700, maybe 1000 subjects, I need to prove that my drug is effective. Here also, the story is simple. How we can prove that whether drug is working or not? Before giving the treatment, we need to collect the data. After giving the treatment, we need to collect the data. Again, by comparing both the data, we can come to a conclusion. But how to do that one? At the end of the day, I need to prove that my XYZ drug is better. My XYZ drug is, do you think your diabetic drug is the first of its kind? Do you think this is the first time that you invented? Do you think there is no diabetic drug in the market? And there are n number of already drugs are available. So, if you want to prove that, what we need to do? We need to prove that my drug is better than some other drug that is available in the market. Maybe my drug is safer than the other drug. My drug is more effective than the other drug. My drug is more, faster than, it works faster than some other drug. So if you want to prove that, well, that means what we need to do, we need to compare our drug with some other drug. By comparison only, we can say that my drug is better or safer or faster or effective. That's why during the phase 2 study, I will take 1000 subjects. I'll divide these 1000 subjects into 2 groups. I'm going to divide these 1000 subjects into 2 groups, 500 into one group, 500 into the second group. To the first 500 members, what I will do, I will give my study drug. By the way, do you know what you mean by study drug? Study drug means the drug which is under investigation. My drug. The drug which is under investigation. My new drug. And to the remaining 500 members, what I will give? I'll give the standard drug. Do you know what you mean by standard? The drug which is already available in the market. Sometimes we also call it as a controlled drug. That means the drug which is going to compare with. Here also same story. From all the 1000 subjects, before the treatment we collect the data. After giving the study drug to these 500 members, we collect the data. After giving the standard treatment to these 500 members, we collect the data. That means in phase 2, how many types of data we are getting? We are getting 3 types of data. Before giving any treatment from all the 1,000, after giving the study drug to first 500, after giving the standard drug to remaining 500 members, you will collect the data. But the most important question is to whom you should give the study treatment, to whom you should give the standard treatment. ఎవరికి స్టడీ ట్రీట్మెంట్ ఇవ్వాలి, ఎవరికి స్టాండర్డ్ ట్రీట్మెంట్ ఇవ్వాలి? You know, on your own, by your wish, we call that treatment as a non-randomized treatment. But to whom I should give this treatment? That what I'm going to do purely based on the chance. Purely based on the chance, randomly, I will select 500 subjects to study treatment. Randomly, I will select 500 subjects to standard treatment. If you are selecting randomly towards a treatment, that concept we call it as what you call it as we call it as randomization.
Randomization.
What do you mean by randomization? If you are selecting a patient or a subject randomly, okay, towards a treatment, is called as a randomization. How we can randomize it? Just like in a lottery method. Just imagine if I have taken a box in that I have put 500 slips of study treatment, 500 slips of standard treatment. If a patient came to the hospital, I'll ask him, take any one slip. He will put a hand in the box, so he will pick one slip. If the slip contains a study treatment, I'll give the study treatment. If the slip has got standard treatment, I'll give the standard drug. Purely based on the chance, randomly. If you select any patient in that way, purely based on the chance, randomly, that whole process is called as a randomization. So, your study is going to be either randomized study or non-randomized study. Non-randomized means we won't do any randomization. Simply, when the subject comes, we're going to give either study treatment or standard treatment. That's it. So, the question that you will get, can you tell me what you mean by randomized study? If you select a subject towards a treatment randomly, purely based on the chance, that is called randomization. In real time, there is going to be a concept called IVRS, Interactive Voice Receiving Recording System. By following, we are going to have few softwares. By using these softwares, we are going to do the randomization. Once you randomize it, now, should I tell the subject that he is going to take the study treatment? Should I tell the subject, reveal that information he is undergoing the standard treatment?
No.
In some studies, we will tell the information to the subject. We let them know. If you let them know about the treatment they are undergoing, that is called as what you call it as open label. Open label means everybody knows, the subject knows, and the doctor knows what treatment he is taking and what treatment he is giving. If you do in that way, what you call it as open label. But if you keep that as a secret,
Blinded.
hide that information, and that concept we call it as blinding. If you mask or hide the information from knowing that what treatment they are undergoing, that concept is called as blinding. And this blinding is generally 3 types. The first one is called as single blind. In the single blind, only the subject is going to get masked or kept as a secret of the subject. The subject is going to get masked, but the investigator will come to know what treatment is giving. By the way, who is the investigator? The investigator is the person who supervises the clinical trial. Generally, maybe a doctor or maybe a research person. So, the person who is taking the treatment is called as a subject. The person who gives the treatment or who supervises the clinical trial is called as an investigator. So, only the subject will be get masked, but the investigator will know that what treatment is giving, and that is called as a single blind. And then we are going to have a double blind. In the double blind concept, both the subject and investigator, the subject and investigator, both is going to get, masked or hidden. Because as investigator, if you know what drug is it is, yes. If I have any sympathy towards a patient, I may give the standard treatment because it is already proven, it is there in the market. So, to avoid unnecessary bias. So what I will do, I go with the double blind. But most of the studies are going to be double blinded. But sometimes we also do triple blind. What will be triple blinding? In the triple blind, both the subject and investigator and the person or a sponsor who supervises the clinical trial or who handles the data, that person also will be get blinded. Because as a sponsor, if I know what treatment it is, as a data handling person, if I know what treatment it is, maybe I may manipulate the data, isn't it? To avoid such things, sometimes we triple blind the study. But most of the studies are either single or double blinded. But now we have a name, fourth wave also, quadruple. In the quadruple, the subject, the investigator, the sponsor, and even the caretaker, the nurse or caretaker who helped the patient also will be get masked, and that is called as a quadruple blinding. So, by following either open label or blinding method, or by following the randomization, we conduct the clinical trial in phase 2. So, at the end of the phase 2 also, tell me what will happen? We are going to have huge amount of data. That's what I can say. Understand this, everybody? Now, once you've completed with the phase 2, then we will move on to the phase 3 study. Remember what was the main focus of the phase 1?
Safety of the
What was the
drug.
main focus of phase? Safety of the drug. What was the main focus of the phase 2?
Efficacy of the drug.
Efficacy of the drug. Now, the main focus of the phase 3 is both safety and efficacy. Both safety and efficacy. But already we have proven safety in the phase 1. We have proven efficacy in the phase 2. Then why should I go with both again safety and efficacy?
Confirmation.
Way to confirm. In the phase 1, we have taken only around maximum 100 patients, 100 subjects. And in the phase 2, we have taken only 1,000 patients. But we need to confirm that one during the phase 3 on a larger amount of patients. Now, during the phase 3, I'm going to select maybe 3,000 to 10,000, even more than that depends on the therapeutic area and disease. We select the subjects, and in these subjects we are going to do clinical trial. When the Covishield, during the phase 3, they have selected 50,000 patients, subjects from India, USA, and Mexico. So, in the phase 3, we take both safe and healthy and diseased patients. So, again, we are going to test the drug one more time on this one. Most of the phase 3 studies are multinational studies. That means, so we are going to conduct these studies in different countries. So, that is one of the reasons why we are going to have a huge subject number. Because if you want to, market our drug in my country, I'll put a condition. You have to test our drug in my population also. So that we conduct the clinical trial. Understand this one in different countries also. So ultimately in the phase 3 also, what you will get now? You will get the data. Okay? In the phase 1 we got the data. In the phase 2 we got the data. In the phase 3 also we got the data. Everywhere we have data and data. Once we collected all the data, what we need to do? We need to do the analysis. As I said, there are around 20 to 25 analysis tools. By using any one tool, I will do the data analysis. Once I did the data analysis, I will get an analysis report. Whatever the report that I have generated, by using this report, now I am going to fill an application called NDA, or we are going to fill an application called BLA. NDA stands for New Drug Application, or BLA stands for Biological Licensing Application. If our drug is only synthetic, a chemical compound, I will fill NDA. But if our drug contains biological substances, a microbial or a biological substance, now we are going to fill BLA application. Anyhow, again, we'll submit this application to again one more time to FDA. Okay, now again FDA is going to take 6 months to 2.5 years time to review your application. They will review your application. Once of review that, if they believe that your drug is very effective, your drug is safe in humans also, then they will give the permission to manufacture and to market the drug. That is how all the pharma companies will get the permission. From this, you know that by following the good manufacturing practices, they will manufacture the drug, then they will market the drug, and they will go to the pharmacies and doctors. They will request them to prescribe the drug, and then they will introduce the drug to the market. That is how they are going to get the profits. Now, everything is a background story. You take any drug that is available right now in the market, it has to go through all this pipeline. You take a Dolo 650 or a paracetamol or a Crocin or diclofenac or isofenac, whatever the treatment drug or surgery that you have considered, it has to go through all this pipeline, then only it will come to the market. Understand this one? Now, what is most interesting in this one? I told you, remember, if you want to market your drug, from where you should get the permission now? You should get the permission from FDA. If you want to get the permission from FDA, what is the application you have to fill?
NDA or
NDA or BLA. If you want to fill an application called NDA or BLA, what you need to have?
data analysis
report. If you want to have an analysis report, what we should do?
data analysis.
We should do the data analysis. We have to do the data analysis. How we can do the data analysis? Now, FDA will ask the question, tell me, what is the tool that you have used to analyze this data, to do the data analysis? Now, according to FDA guidelines, to do the data analysis, clinical data analysis, you have to use only SAS. That's why you guys have learned SAS. Understand this one? According to FDA guidelines, the clinical data or clinical study data, we should analyze only by using SAS. Why we should use SAS? Why FDA recommends only SAS? That's the question. Isn't it? Why FDA recommends SAS? Because SAS has got two advantages. Very good. The first important advantage is data safety and security. No other tool can provide this much of data safety and security. This is one of the reasons why FDA recommends only SAS. And the second important condition advantage is SAS is called a complete analysis tool. What will be a complete analysis tool? By using SAS, we can do data access, we can do data management, we can do data analysis, and we can do data presentation or data reporting. No other tool can do everything at one place. So this is the second reason why FDA recommends only SAS. It is recommending from last 30-40 years, and they are associating now also. Understand this, everybody? Now, sometimes FDA will give the permission to market the drug, but if they'll put a condition, once a drug went into the market, collect the data from the hospitals and again the patients. And collect the data, analyze the data, and again submit the data. So that means once the drug went into the market also, because FDA wants to see what is the long-term effect of the drug. Because sometimes a drug, maybe if you are using drug for 1 month or 2 months, may not show any side effects. But if you use the same drug for longer duration, it may show some side effects, isn't it? So now they want to know the long-term effect. So to understand that long-term effect, they ask you to submit the data even once the drug went into the market also. And that we call it as an A. What you call it as an A? Phase 4 studies, also call it as an A, post-marketing surveillance. Post-marketing surveillance.
Surveillance.
Because they want to know that one. Understand this one? So these are the 4 phases of clinical trials. We have in humans. If a clinical trial that is not safe and secure, people like me may manipulate the data. If FDA gives the permission based on the manipulated data, ultimately it is going to affect the human health. In the past, there were such incidences. That's why FDA is very strict when it comes with the data safety and security. Understand this for everybody? Now, you came to know one very important thing. In clinical domain, SAS is mandatory. According to the FDA guidelines, we should use only SAS. Since we should use only SAS, now you guys have learned SAS. Now, I go back to the let's say for example, previously, all the pharma companies used to submit that analysis report to FDA. But when they're submitting the pharma companies their reports to FDA, each pharma company used to submit the report in their own manner, in their own way. Because of that, FDA used to take so much of time to review the application because they never know how the report is going to come. Once the report comes, they have to train FDA people to review the application. Because of this, the pharma companies used to lose so much of time. Remember I told you, when you discover the drug in the starting, how many years you'll get the patent?
20 years. Yes,
20 years. To complete the R&D, initial preclinical studies, in vitro studies, in vivo studies, then phase 1, phase 2, phase 3, to get the drug into the market, it will take 10 to 12 years time. It will take 10 to 12 years. That means already you lost 10 years out of 20. In the remaining 8 to 10 years, you have to market our drug very effectively. Then only you will get the profits. Otherwise, if you didn't develop the drug further, after the 20 years, you lose the patent. So anybody can now manufacture this drug now. So that is called a generic. If the generic drug comes into the market, nobody will buy from you because the generic drug, you didn't do any research. What is a generic drug? After 20 years, whatever the research whatever the tablet that you have manufactured, what I will do, I will take this tablet to my house. I'm going to grind, I'm going to make this powder. Once it becomes powder, I'm going to separate all the chemical components and I will measure how much components that we have in each tablet. Again, what I'm going to do, I'm going to mix all the components in large scale, bulk scale. Again, I'm going to manufacture that, I'm going to create the tablets and I get that into market. Whatever the tablet that you prepare, this is also going to be the same composition. But you need so much of research, so much of money that you spend. If you want to market this drug for your tablet, it may take 5, it may be cost you 10 rupees for you to get a profit. But since I did a reverse manufacturing process, it will going to cost me only 2 rupees. If I sell my product even for 5 rupees also, I'll get 150% profits. I didn't do any research. That's how we get the generic drugs into the market. If the generic drugs comes, you lose your profit. So in clinical domain, time is so important. Time is money. Time is money. And that too, remember, I told you in the starting, if you think 10,000 molecules has got potentiality, you think the 10,000... Could you please turn off your mic? If you think 10,000 molecules has got potentiality to get, how many drugs is going to come to the market? 5 to 10 drugs will come into the market after the phase 3 study. Can you see the success ratio? 10,000 is to 5. The success is very, very minute. So much of money involved, so much of time involved, so much of efforts involved. That's why all these pharma companies will take lot of care in each and every step. How much care that you take after aviation industry. Right, what you mean by aviation industry? All these aeroplanes, passenger, and all these. So in the aeroplanes, aviation industry, accuracy is very, very important. If there is even small error is also, it is going to become a big disaster. So after aviation industry, we follow that much of accuracy, we take that much of care only in clinical research. Because every step is very important, every data point is very, very important. So the success is very bleak, and so much of money, so much of time involved process. Every pharma company will take lot of effort to make sure everything is correct. But as I said previously, pharma companies used to take a lot of time because FDA, they doesn't know how the report is going to come. So what they used to do, once the report comes to them, then they have to prepare the FDA personnel to review that application. Because of that, the pharma companies used to lose so much of time. Because of that, now all the pharma companies decided to submit the reports in the uniform manner. How to submit the reports to uniform manner to FDA? Now, all these pharma companies, now they have created an independent, open, multinational organization called CDISC. What CDISC will do? The CDISC will go to the FDA and they will ask in which way you want to have the report. What you are expecting. So in which way you want to have the report. If the pharma companies want to submit the report in that manner, what are the guidelines they need to follow? Now, this CDISC is a multinational non-profit voluntary organization. Now the CDISC will frame the guidelines. It will frame the guidelines. Now, all these pharma companies, by following the CDISC guidelines, they will submit their analysis report and data to FDA. That's how now the pharma companies are following the CDISC guidelines. But still, CDISC guidelines are not mandatory. If a pharma company wants to submit the report in their own manner, still they can submit to FDA. But FDA will take a lot of time to review that application. So pharma company doesn't want to lose the time. So that's why voluntarily all the pharma companies will follow the CDISC guidelines. So that means CDISC works between the pharma companies and the FDA. But FDA is what? It's a USA body. And in India, if you want to get a permission, if you want to market the drug in India, you need to get a permission from DCGI. If you want to market drug in Japan, we need to get a permission from PMDA. Each and every country is going to have a regulatory body. But one country's regulatory body may have a different guidelines from the different another country. Now, to make it simple, to make it uniform between the countries' regulations, now there is going to be one more organization called, what that organization? We have an organization called ICH. The ICH is going to make sure having a harmony between different country guidelines. So, CDISC works between pharma companies and FDA, and ICH works between the country's regulatory guidelines. Understand this one, everybody? Now, CDSC has framed two important guidelines. One is SDTM and the second one is ADAM. When you're submitting the clinical trial data, what are the guidelines you need to follow? They have framed some guidelines. Those are called SDTM and ADAM. By following these guidelines, pharma companies will submit their clinical study data to FDA. Understand this one? The first 1.5 months, we are going to learn about STDM, and then after that, we will learn ADAMS, and then after that, we will learn about data analysis. In the data analysis, we are going to have tables, listings, and graphs, nothing but figures. Is this clear, everybody?
Sir.
So, in tomorrow's class, I'm going to tell you what is this STDM and ADAMS, how we conducted each clinical trial phase, how we conduct a phase 1 study or a phase 2 or a phase 3, how we collect the data, and then how we start with the SGDM, then ADAMS. Clear, everybody?
Sir.
Anybody has got any doubts so far? If you do not have any doubts, I'm going to stop this class here for today. We'll continue in tomorrow's class. Okay? Does that sound good?
Yes, sir.
Yes,
Okay
sir.
then.
Yes, sir.
Okay guys then. See you in tomorrow's class.
Thank you, sir.